Good evening, everybody. Hope everybody is doing well in this unforeseen pandemic outbreak and a subsequent lockdown period. Our biotechnology department wants to keep our people educated towards ongoing research and towards career enhancement, etc. In this regard, the Department of Biotechnology is hosting a special webinar series called Biotalk Lecture Series. This is the second week event actually. As the participants are from various places, I wish to brief some of the highlights about our college and the department. St. Joseph College of Engineering is one among the top engineering colleges in India with the 160th position as per current NIRF ranking. It has got 26 years of high reputation in educational society with the NAC A plus grade accreditation. It is having 10 UG programs and 7 PG programs with an affiliation from Anna University, Chennai. Our Department of Biotechnology, is 18 years young and having BTech and MTech biotechnology programs. So far, we have bagged 139 university ranks with 10 golds and 7 silver medals with 100% pass percentage. The department has got high-end sophisticated laboratory facilities and fetched a research center recognition status from Anna University to do PhD program and having more than 1.1 crores of rupees of government funding to conduct research. The disciplines such as electronics, computer science, information technology, etc. touches the lifestyle of the mankind, but the biotechnology alone touches the life of the mankind. Hence, an in-depth knowledge in any of the biotech field becomes mandatory to do biotech research. In other words, a post-graduation is a must in the field, unlike other disciplines. By keeping all these things in mind, Department of Biotechnology has decided to organize this week's Biotalk lecture series in the topic of bioscience research and master's opportunity in UK. For this, we have Dr. Pranav V and Mr. Nidhi S from Scots Hunt UK Limited and Scots Grant. Dr. Pranav is an alumnus of St. Joseph College of Engineering, Department of Biotechnology. I'm proud to say that. He's a senior research fellow in the field of biological engineering. He has got Masters in Immunology, PhD in Biomedical Engineering Research Group, and he has got the first postdoctoral research in chemical engineering and the second research fellow position at the University of Birmingham. So he is working as a cross-disciplinary researcher for the projects that require knowledge and application of physical and engineering principle to solve complex biological problems. They would be definitely sharing their views of biological research and higher studies opportunities in UK. At this moment, I wish to comment about our participants also. There are around 120 registered participants from various colleges and universities, both at the UG and PG level. With this proud note, on behalf of the patrons of the college, Dr. Babu Manohar, Chairman, Madam Jessie Priya, Managing Director, and Mr. Sashi Shekhar, Director, Dr. V. Seshagiri Rao, Principal, and Dr. Parvatavardhini, Dean's Madam. I welcome these invited speakers and all the other participants to this BioTalk lecture series. I take this opportunity to convey my warm felicitation to the organizer, Dr. G. Bhaskar and Madam Renuka from St. Joseph, and a special mention to Mr. Nidhi and Mr. Kadiravan, Executive Director, Scots and UK Limited. And I extend my best wishes for the success of this webinar. I'm sure that this will definitely provide an excellent opportunity to all the participants to get information about biotech research and higher studies opportunities in UK. All the best. Thank you. Over to Dr. Pranav.
So yeah, thank you so much, Professor Dr. Sri Kumar, for kindly accepting me to be a part of this webinar. And thanks also for Nidhi and Kadir for organizing this wonderful webinar, which I'm pretty sure is going to stimulate a lot of interest among the young aspiring scientists who are listening to this. So the idea of my presentation today is to just give a brief overview of the sort of projects I've been. Okay, so let me just briefly go through my research career so far. So I started off by doing my bachelor's in biotechnology from St. Joseph's College of Engineering in Chennai. And I'm so proud to be an alumni of this institution. It was a wonderful course with several different elements from chemical engineering, biotechnology, immunology, etc. But the subject that fascinated me the most was obviously immunology. So I definitely wanted to do my master's in immunology, and upon doing my research, I found that UK provides a variety of options in this subject. So I packed my bags. I came to the UK to do my master's in immunology and allergy from the University of Nottingham, which is one of the top 100 institutions in the world. And it was a phenomenal course. We literally sprinted through the course because, as you would know, masters in the UK are like a 1 year course. So it's highly compact and rigid, and but it also offers several possibilities. So one of the advantages of a master's in UK is that if you are very keen on doing a PhD, you can save an entire year's worth of time and money compared to 2 years or 1.5 years masters of other countries. So towards the end of my masters, I was very keen um, on doing my PhD in tissue engineering. Tissue engineering, as you know, is an area of research which develops complex in vitro models of several human diseases. Now, I also wanted to be a bit more experimental in my PhD. So not just being a strict biologist, but also a sort of a bioengineer. So despite having a master's in immunology, I took a leap and decided to do my PhD in mechanical engineering, right? So which is a very uncommon move. Yeah, I decided to do my PhD in mechanical engineering, and which is a very uncommon move among the biologists because a biologist wants to specialize within a biology stream. Um, but I was really interested in applying engineering principles to solve biology problems. And my PhD involved equal elements of biology and engineering. So my PhD, which I'm going to talk about in detail in a few slides, very briefly was to study the health effects of electronic cigarettes. And this involved developing in vitro models of the human airways, as well as building a bespoke smoking robot that would mimic the human smoking behavior. So one thing led to another, and after completing my PhD, I wanted to do my postdoc uh, in a in a very unique field called organ on a chip or tissue microfluidics. So my first position was again another risky move wherein it was a 5-month project, but as you would know, if you really have a passion for science and if you demonstrate some really good work, you can get a lot of work done in 5 months. And one thing led to another, and I got a really good position at the end of my 5 months to be a part of this big consortium project at the University of Birmingham. Can we go on to the next slide? So I'm going to just briefly go through some of the projects which I've been involved with. And one of them being my master's research project. So I started off by doing, as I said, my master's in immunology. And so let me just start off by talking about my master's research project. So the idea was to investigate biocompatible properties of a novel hydrogel called gelatin methacrylate. Now, hydrogels, as you might know, are this attractive class of biomaterials that are used in several different applications like tissue engineering, drug delivery, as well as regenerative medicine. We wanted to specifically investigate how the immune cells react to these sort of hydrogels. So peripheral blood mononuclear cells were isolated from very kind donors, and they were cultured on top of these gelatin methacrylate hydrogels, and the cell viability and cytokine profiles were analyzed. We also tried to investigate if the biocompatibility of this class of hydrogels can be improved by adding additional ECM ligands such as collagen fibronectin. Next slide. So, sorry. So going back to this preparation of gelatin methacrylate, now a biologist would know that gelatin is actually nothing but hydrolyzed form of collagen, which is the most abundant protein in the human body. So collagen was extracted from porcine skin and hydrolyzed to form gelatin. And to that, we add a chemical called methacrylate anhydride. Now, when these two chemicals, when methacrylate anhydride is mixed to gelatin, it's chemically modified to have these methacrylate side chains, which in the presence of a photoinitiator and under the exposure to UV, they cross-link, and depending on the time of UV exposure and the concentration of photoinitiator, you sort of get soft to stiff hydrogels, depending on the purpose for which you want to use it to. Now, another advantage of using this method is that UV irradiation sterilizes the hydrogel and hence making it more biocompatible for cell culture. Next slide. Right, so this is how an average day in my lab would look like. So this is a very common experiment which I did during my master's. So just going through this plate layout, this is a 24-well plate, and in each row, you've got different combinations of gelma. So gelma plus collagen at different concentrations, gelma plus fibronectin, gelma plus collagen and fibronectin, gelma control alone, and tissue culture control, which is cells growing on the plastic surface. Next slide. So one of the key things which a biologist wants to know at the very first hand is whether the cells are happy on top of any substrate. So we wanted to check if the cell viability is compromised when cultured on top of gelma. And as you can see, they're very much comparable to the cells growing on tissue culture control. So the cells were very happy, and the percentage of apoptosis that was investigated using annexin V FITC fluorescent molecules were demonstrated that about 70% of the cells were alive on gelma. In the next slide, what I'm going to show you is we investigated the four key pro-inflammatory mediators, namely TNF-alpha, IL-1 beta, IL-6, and IL-8. Now, this was a very key result in the project because, as you can see, in the presence of LPS, which is a bacterial endotoxin, which would stimulate fierce production of pro-inflammatory mediators, you can see significant increases in the tissue culture grown cells. Whereas in the gelma, there wasn't much difference between LPS plus LPS or minus LPS, which shows that gelma sort of acts as a sponge, which sort of soaks up these pro-inflammatory mediators that are produced. And this is a very key result because it has significant implications in terms of in vivo transplants of gelma, where there's excessive amount of pro-inflammatory mediators production, as seen in asthma or COPD or ulcerative colitis. We could probably use these materials as delivery agents. Now, in the next slide, you'd probably, this is a validation experiment where we did conventional gel electrophoresis and qPCR to validate the results observed in the previous slide. As you can see in the 30 minutes timeline, there's TNF-alpha production both on tissue culture plates as well as gelma, but after 4 hours, the gelma the TNF-alpha in gelma band sort of disappears, which definitely shows that they have been soaked up by gelma. And that the very same result was further verified by qPCR. And on your result on the right-hand side, you can see that TNF-alpha, without any cells, when added to the gelma, they were being absorbed by the hydrogel compared to the tissue culture plate. So in the next slide, I'm going to show you I'm going to encourage you to go and read this paper, which was published. It was a teamwork between University of Nottingham, Harvard University, as well as a number of other departments within Nottingham. And you can find a lot of details, which I'm not able to talk about right now due to shortage of time. So we move on to my PhD project. I call it the big challenge, and you'll know why in a couple of slides down the lane. So my PhD project was a cross-disciplinary project with essential elements of both tissue culture cell biology, as well as computer science, mechanical engineering, and electrical engineering. It was a very, very complex and a challenging project. The idea was to investigate a very hot topic, which is widely discussed across the globe, e-cigarette cytotoxicity, using physiologically relevant in vitro methodologies. Now, why did I choose in vitro methodologies? Because despite the fact that animals are widely used, the the airway architecture between the species is widely varied, and hence the results that are obtained from mice cannot be translated into any therapy for airway conditions. So we want to move very much towards in vitro, more physiologically relevant methodologies. So what were the research questions that, sorry, can we go back to the previous slide? So 3 research questions. What cell model can I employ that would represent the in vivo human conducting airways environment as closely as possible? How can I deliver cigarette or e-cigarette aerosol in a fashion mimicking human smoking behavior? And lastly, the most important question, how does e-cigarette aerosol compare to cigarette smoke, and what are the factors that influence the cytotoxicity? Now, in the next slide, I'm going to show you the cell types that I worked with. A variety of cell types were used, chiefly bronchial epithelial cell type, immune cell types, as well as fibroblastic cell types. But the 2 chief cell lines that I used, which would be highlighted, are Calu3 bronchial epithelial cells and MRC5 fibroblasts. Now, in the next, yeah. Now, in the next slide, I'm going to show you how did I produce this co-culture model of human airways. I used transparent permeable polycarbonate membranes, which are commercially called transwells or snapwells. I insert them in a 6-well plate. I supply them with growth medium from the bottom, and I coat them with type 4 collagen, human type 4 collagen, which are abundant in the human airway architecture. And can you press? So what I did was the red cells, which are the pulmonary fibroblasts, I culture them under submerged conditions for about 4 days, after which I add the bronchial epithelial cells on top of these fibroblasts, and I culture them, these 2 together under submerged conditions for another 4 days so that they grow, expand, and proliferate. And on the 8th day, I remove the air medium from the apical layer, and I expose them to growth medium from the bottom, thus establishing an air-liquid interface. Now, such a condition would lead to the production of mucus-producing goblet cells, tight junction formation, as well as cilia production. I then expose to cigarette smoke or e-cigarette vapor using an interesting engineering design, which I'm going to show you in the next slide. So again, before going to the engineering design, one of the important things a biologist should learn is to characterize their cell model. So what did we do to characterize this? We did immunofluorescence, which chiefly looks at the epithelial markers CK5 and CK8, as well as the fibroblastic markers, namely IB10. We also took some interesting scanning electron microscope pictures that depicted both the fibroblastic layer as well as the epithelial cell layer, and it depicts the tight junction formation, microvilli production, as well as the goblet cells production. So in the next slide is where I introduce the engineering aspect of my project. Now, how can I deliver cigarette smoke or e-cigarette vapor in a fashion that mimics the human smoking behavior as closely as possible? There is a standard for this, an ISO standard called 3308, and that would act as an inspiration for this particular design that we developed, which would pretty much set all the parameters of the human smoking behavior. So in the next slide, I can show you an overview of this particular design. Now, although there's a lot of things that are going on in this slide, and it looks very complex, I'm going to make it a bit more easier for you to understand. So you can see the cigarette holder in the middle. So you insert your cigarette or e-cigarette, whichever you want in there, and you place your core culture in vitro models in these transparent perspex blocks, and you connect them to the smoking machine, which was designed by myself and a couple of other undergraduate students. Now, what would happen is for 58 seconds, just normal air from the air would be delivered to the co-culture model. And for 2 seconds, cigarette smoke or e-cigarette vapor would be delivered to the same model. And these two processes would alternate with each other. And you can literally play around with the amount of puffs you want to expose your co-culture model to and the amount of time you want them to expose them to, because individual cigarette smokers have their own patterns. Now I'm going to show you in the next slide how it looks like in real time. This is a literal real-time translation of the previous slide, and it was almost a year and a half work to build this entire machine. I'm sure you can appreciate that being a biologist coming, doing biotechnology and then immunology, this was a very challenging task for me to learn, electronics and microcomputing and mechanical engineering, apart from also developing the co-culture models. Next slide. And then the cells were then exposed to cigarette smoke or e-cigarette vapor at several different time points. So 1 hour to 6 hours at a rate of 1 puffs per minute. After that, we performed a number of biological and molecular biology assays, such as cell viability, IL-6 and IL-8 production using ELISA. We used a fluorescence technique to measure the oxidative stress, and we used a similar fluorescence techniques to measure caspase 3 and 7 release, and we employed RT-PCR for BAX BCL2 gene expression levels. I'm going to encourage you to go and read this paper that was published in Toxicology in Vitro. And if you press the next slide, Kadir. And so at the end of my PhD, as I said in the beginning, I was very keen on performing, transitioning into this very nascent field called organ on a chip or microfluidics technology. It's a very nascent technology, about a decade and a half old. And my idea was to sort of stick with human airway conditions. So I wanted to investigate what are the mechanobiological problems associated with human airway fibrosis. Now, what do I mean? As you may know, fibrosis is the condition where the extracellular matrix on which the cells are residing, they become hardened, and they sort of get calcified in a manner in which the cells start degrading, and they would undergo a process called epithelial to mesenchymal transition, which would then lead to a vicious triad of cancer, abnormal wound healing, and fibrosis. So to investigate this triad of cancer, wound healing, and fibrosis, I started off with fibrosis. And the research question that I had was, do the mechanical coups, by which I mean the underlying extracellular matrix, do they override the influence of biochemical signals such as cytokine growth factor during fibrosis? Second question was, to what extent do epithelial cells contribute to fibroblast to myofibroblast transition in airway fibrosis? And the third most important question was, can I target the integrin or the GPCR intracellular pathways, which are SMAD pathway and Wnt pathway? Can I use these pathway to reverse the pro-fibrotic phenotype, or at least, to stop fibrosis from becoming cancerous or aberrant wound healing? Next slide. So a very simplified model, a very simplified model of what I did was, I used state-of-art microfluidic techniques, and I used a physical principle called thermophoresis, which is the movement of solutes in a solvent upon application of a temperature gradient. The solutes move from low temperature to high temperature, or from high temperature to low temperature, thus forming a concentration gradient. So as you can see, this is a strip of hydrogel that was obtained from a microfluidic model. And what this hydrogel has is, across its length, it ranges from soft to stiff region. So the softer region are the regions where the cells would reside in a normal ECM matrix in vivo, and the stiffer regions are where the cells would reside in a fibrotic ECM matrix in vivo. So I wanted to understand how the cells migrate and differentiate between the soft and the stiffer regions. Especially and specifically, I wanted to understand the process of epithelial to mesenchymal transition and fibroblast to myofibroblast transition. Next slide. So in the next slide, I'm going to show you the exact process that I did. So bronchial epithelial cells, namely Bs to B cells, about 30,000 of them were added on top of this hydrogel, and they were incubated for about 7 days. On the 7th day, I then removed the growth medium. I fixed them with paraformaldehyde and tried to next to permeabilize the cell membrane, and I perform immunofluorescence using specific markers for epithelial cells, which is E-cadherin, fibroblast, which is vimentin, and myofibroblast, which is alpha SMA. Now, bear in mind that if you detect that a lot of alpha SMA in your samples, it means that the fibroblasts, that the cells, the epithelial cells have actually undergone fibroblastic transition, and fibroblasts have undergone myofibroblastic transition. Next slide, I'm going to show you pictures of immunofluorescence that was obtained. So as you can see over here, this is my hydrogel. It ranges from soft to stiff, and you can see that the cells in the softer region are more clumped together. They form they have significant tight junction formation, whereas towards the stiffer region, they sort of elongate and they exhibit more fibroblastic phenotype. A magnification of this particular image shows us that there is a lot of expression of vimentin along the cell periphery, which shows that there has indeed been a transition from epithelial to mesenchymal transition. In the next slide, I'm going to show you two other additional images of uh, epithelial to mesenchymal transition. So as you can see over here in the stiffer region, the vimentin production is more predominant towards the cell periphery, whereas in the softer region, you can see that the cells are more clumped together, and also the vimentin is all across the cytoplasm and not just at the cell periphery. So this means that stiffer region actually are influence the cell biology, the production of actin cytoskeleton and vimentin production, and that leads to, that exacerbates the fibrotic condition. In the next slide. So together, summing up all my skills that I've gained, I have got significant experience in employing hydrogels for tissue engineering. I'm adept at both 2D and 3D cell culturing. By 3D, I actually mean proliferation of cells within three-dimensional matrix. As you would have seen in my in vitro co-culture model of human airways, as I showed you, I've learned how to validate these cell culture models using relevant markers. And here's an interesting bit. If you're a biologist and if you want to create a niche, I would highly encourage any enthusiastic biologists to also start developing some engineering skills as well. And this would create a very niche profile for yourself. So apart from immunology and other cell biology experiences, I also have experience. I've also learned building bespoke machines like the smoking robot, microfluidic techniques, and also currently learning in silico models, which are mathematical modeling of complex conditions that happen in the human body. So I'd like to acknowledge all the people that I've come across in my next slide: Professor Amir Gamigami from Nottingham, Amy Donaldson from Nottingham, Laura Leslie and Lindsay Marshall, James Brown from Aston, Daniele Vigolo, Mark Simmons, and Liam Grover from University of Birmingham. It wouldn't have been possible without these people, and I'd very much like to acknowledge their presence in my research career. So that brings me to the end of my research journey. And in my next slide, what I'm going to do is I'm going to go through the several different options for the youngsters, which are possible upon coming to the UK to do their masters. So can we go to the next slide, Karan?
Yeah, Pranav.
So hang on. So if you come here to do your masters, either a taught masters or a masters by research, what you can do is you can choose to do a very traditional, undertake a very traditional pathway by choosing to do your PhD and then doing a couple of postdocs across different lab, enhance your publication numbers and then choose to become a lecturer. Now, this is a very, very traditional pathway that's been there for hundreds and hundreds of years. But what you can also do now that the post-study work visa has opened up, you can apply for internships or start off as the junior scientist at several leading industry in the UK, such as GlaxoSmithKline or uh, Novartis or AstraZeneca, and you can gain a lot of experience as a junior scientist, and then you can transition towards becoming a more senior scientist, which is generally a permanent position. Now, an interesting thing, what you can do is if you're unsure whether you want to stay within academia or whether you want to go into industry, you can also choose to do a research assistant position. Can you click on Kadir? So a research assistant is nothing but it's a intermediate between a master's and a PhD. So you can actually work in the labs of major principal investigators, and you can gain hands-on experience within the lab, and you can then decide whether you want to do research within a university setting or you want to work within industries. Uh, what are the alternative research career pathways? If you're one of those personalities who loves taking risk and enjoys a high octane environment, then you can also choose to apply for a tier 1 visa in terms of entrepreneurial ventures. There are huge number of funding options. I'm just going to talk about two over here: Innovate UK and Crack It from NC3Rs. But bear in mind, this tier 1 option, this is not just restricted to, like, the smartest of the smartest or the brightest. The UK government actually encourages a lot of foreigners, a lot of immigrants to apply for this particular position because it works well for their economy and it works well for you as well. Now, you can also choose to work for the British Home Office or Science Cancer Research UK or British Heart Foundation, which are charities. And bear in mind that you can work within the British Home Office despite not being a resident. It is not restricted to just the local British people. Now, one of the other interesting pathway which you can choose to do is to do science communication. So you can start off by working for any of these charities: British Heart Foundation, British Lung Foundation, Cancer Research UK. You can write regular blogs being a part of them, or you can set up your own YouTube channel and engage with the public as much as possible. This is a very up and coming stream, and a lot of youngsters are engaging in this particular form of research career. It's quite a good paying. In fact, you can earn as much as a lecturer by doing this. And another subset of the science communication is to combine science and art. I'm pretty sure there are a lot of people who enjoy science, but they also they're very creative and they enjoy art as well. So, for example, I have one of my friends where she used to take brilliant pictures of cell membranes and nucleus under the microscope, and she used to print it out, edit it and print it out on coffee mugs and T-shirts, and she now has a brand of herself. So this is you can do a variety of options once you start yourself with a master's in the UK. So that essentially brings me to the end of my presentation. I'm pretty sure there's a ton of questions which many of you might have, and I'm more than happy to interact with the individual questions of the audience. Or if there's a shortage of time, Kadir and Nidhi would curate them, and I would be very happy to interact with individual members upon a later stage. Thank you so much.